Enzyme reactions have to be controlled. This is important so that some reactions do not happen too quickly, which could result in the build-up of potentially dangerous levels of excess products. Enzymes also help regulate vital metabolic reactions, and it's vital that these occur at the necessary level without wasting resources. Today we're going to look at the different types of enzyme inhibition: competitive, non-competitive, and end product. Firstly, let's look at competitive inhibition. Here, a molecule or part of a molecule that has a similar shape to the substrate of an enzyme can fit within the active site of the enzyme. This will block the substrate from entering the active site, preventing the enzyme from catalyzing the reaction. The enzyme cannot carry out its function and is said to be inhibited. The non-substrate molecule that binds to the active site is a type of inhibitor. The substrate and the inhibitor molecule will compete with each other to bind to the active site, and this will therefore slow down the rate of reaction. We call these inhibitors competitive inhibitors, and the degree of inhibition will depend on the concentration of all of the involved molecules. Most competitive inhibitors only bind temporarily to the active site, so their effect is reversible. There are, however, some exceptions, such as aspirin. Secondly, we'll move on to non-competitive inhibitors. Here, the inhibitor binds to the enzyme at a location other than the active site. This alternative site is called an allosteric site. The binding of the inhibitor causes the tertiary structure of the enzyme to change, meaning the active site changes shape. This results in the active site no longer being complementary in shape to the substrate, so it's unable to bind to the enzyme. The enzyme can't carry out its function, and it's said to be inhibited. Both of these enzyme inhibitors will affect the rate of reaction. In a non-competitive inhibitor, increasing the concentration of either enzyme or substrate will not overcome its effect. Increasing the concentration of the inhibitor, however, will decrease the rate of reaction further, as more active sites become unavailable. A competitive inhibitor will reduce the rate of reaction for a given concentration of substrate, but it will not change the Vmax of the enzyme it inhibits. Increasing the substrate concentration will mean there is more substrate than inhibitor, so the Vmax. Here you can see the earlier information represented on a graph of substrate concentration against rate of reaction. The yellow line represents the reaction taking place without any inhibitors, and you can see that where it levels off it reaches its Vmax. Were we to add a fixed quantity of competitive inhibitor, you can see that rate of reaction slows, however the reaction still reaches its Vmax. The orange line shows a fixed quantity of non-competitive inhibitor being added. Again, you can see the rate of reaction slows, but more importantly, the reaction does not reach Vmax. The last type of inhibition we'll look at is end product inhibition, and this is used when the product of a reaction acts as an inhibitor to the enzyme that produces it. It's an example of negative feedback. Here you can see that as the substrate is broken down into the products, one of those products binds non-competitively to the enzyme, changing the shape of the active site. This stops any further reaction. This is important as it ensures that the excess products are not made and resources are not wasted. It's an example of a non-competitive reversible inhibition. An example of this can be seen in respiration. Respiration is a metabolic pathway that results in the production of ATP. Glucose is broken down via a number of steps to produce the ATP. The first step involves the addition of two phosphate groups to the glucose. The addition of the second results in the initial breakdown of the glucose molecule, and this step is catalyzed by the enzyme PFK. This enzyme is competitively inhibited by ATP, and therefore ATP is regulating its own production. When ATP levels are increased, more ATP is available to bind to the allosteric site on the PFK. This prevents the addition of the second phosphate group to the glucose, and therefore the glucose isn't broken down and ATP is not produced at the same rate. However, as the ATP is being used up, less of it is available to bind to the PFK, and the enzyme is able to catalyze the addition of a second phosphate group to the glucose. Respiration will therefore resume, and this will lead to the increased production of more ATP.